Hello, Genetics 311 students. Welcome back to my channel. Today we will be talking about synapsis of chromosomes. So synapsis is an event of meiosis 1 that occurs in prophase 1 and is when homologous chromosomes pair with their counterparts and they remain bound to each other due to the exchange of DNA. And the functions of synapsis is to hold the homologous chromosomes together and exchange information through crossing over. We'll begin talking about synapsis through individual 1. Looking at these chromosomes, there is a pericentric inversion. So this occurs when the inverted area includes the centromere. Looking at our inversion loop, we see that the crossover event happens between J and K. So the gametes that it produces are as stated right here, and there's only 2 viable because it contains all of the different alleles. So there is no loss of DNA, whereas here there's a deletion because it doesn't contain all of them. Looking at individual 2, the chromosomes that we are presented with are the exact same. If you just flip this one the other way, it can be read backwards and forwards, the same as this. So there won't be an inversion loop in this case, since it's just going to be a simple crossover event. But, as said earlier, they are the exact same, so they will all be copies of the parent. This means that all of them will be viable, and there will be a higher fitness than individual 1, since it only produces 2 viable gametes compared to 4 in this case.